Now it's my pleasure to introduce Dr. Behnam Sadeghi, a highly respected expert in the field of regenerative medicine and cell therapy. He currently serves as Senior Scientist, Head of Translational Cell Therapy Research TCR at Department of Cleantech, Karolinska Institute in Sweden, and Chief Scientific Officer of Taskin BioRegeneration. Dr. Sadeghi work focused on bringing cell-based treatment from research into real clinical use, and his research and leadership are helping shape the future of how we treat disease using advanced therapies. Dr. Sadeghi, we are honored to have you, and the screen is for you. Thank you.
Thank you very much for inviting me to join this valued meeting. I will start my presentation by this. So next generation of cell therapy, deciduous stromal cell for acute inflammatory disease. As you may know, the acute inflammatory diseases are a huge part of the disease among human beings, and they start from the iatrogenic disorder like GVHD, ARDS, until several natural inflammation like sepsis, lupus nephritis, and myocardial infarction. The problem with all of these diseases, first of all, they are very complex, the pathophysiology, but very similar to each other. And the second thing which make them common together is that limited therapeutic options. However, these limited options has a very high side effect and also high cost, as well as a very negative impact on the quality of life of the patient. Therefore, there is an unmet demand for the new and effective drug treatment. Many years ago, stromal cells were introduced to the clinic, to the scientific field, and as a research, and everybody knows that these cells comes from the bone marrow, and they can differentiate to the cartilage, bone, muscles, and later on, a lot of scientists found them in different tissue from adipose, umbilical cord, placenta, bone marrow, as well as a lot of different organs. We at Karolinska Institute were the first group in the world who introduced bone marrow MSC for the treatment of the first case of steroid refractory GVHD in the world. It was a baby who got the MSC from the mother, haploidentical MSC. And this boy was responded very well, and after 2 doses, the patient recovered and discharged from the hospital. And later on, we did another study. We noticed that, yes, those who respond have better survival compared to those who do not respond. The big question mark came from here, why some people are responding and one not responding. After our first experiences, a lot of studies started worldwide using MSC in a lot of different modality, which the reason could be different, some of them degenerative disorders, some of them inflammatory disorder. Very soon we noticed that this is not a good approach. It was a wrong. The same problem happened even for treating the SR-GVHD by the MSC, the first disease that was introduced to using the MSC. Parenchymal or mesoblast nowadays, they failed to reach to the primary endpoint in the study. Therefore, the FDA reject their application. Again, we noticed that we did another control study at our institute. We noticed that the patient with and without MSC treatment have a similar survival. The same observation was repeated by Kebriai teams in a phase 3 randomized trial worldwide. Therefore, this mesoblast could not get the FDA approval, even withdraw from the New Zealand and Australia. And later on, a Cochrane Library paper came up and mentioned that the bone marrow MSC is not effective for the treatment or prevention of the GVHD. What should we do? Among all of the cell therapy or stromal cell base, we noticed that the base model of tolerance induction and immune modulation is a pregnancy. A haploidentical baby always is protected from the mother immune system. The reason is because of these very nice, beautiful cells which are leaning in the placenta as well as the membrane. by a very dedicated enzymatic method, we could manage to isolate these cells. The journey starts from here. When I'm talking about the steroid refractory GVHD, I mean this type of the patient. They are almost dead. The mortality is more than 95%, even by the using of a steroid or corticosteroid. So as you can see in the blue line, you see the survival of SR-GVHD without any further or second line treatment because at that time this did not exist. So now when we are giving this patient, blue color, DSC, you see that the survival increased to the level of the normal patient. It was too good. When we submit this paper, the reviewer said that too good to be true, but finally they accepted, and this paper was the highest cited in the 2018. Imagine the efficacy of the bone marrow MSC versus deciduous stromal cell or DSC, which is professionally anti-inflammatory. And then in the lab, we compare the immunomodulatory or prevention of the allogenic lymphocyte proliferation among the FMC, which was the first name of DSC, versus umbilical cord, placenta villi, and the bone marrow. And as you can see, the immunosuppression is 3 times to 5 times more compared to the bone marrow MSC. So one thing which is very important is the origin of these cells. They come from the placenta, which is evolutionary for immune modulation or immune tolerance induction, while the bone marrow comes from the bone marrow and is helping the hematopoietic cells. They have several differences. Because of the shortage of time, I love to move forward for the last message. So why the BDSC is much better. The first is that it is the best-in-class immunomodulation based on the evolutionary. Naturally, they induce the immune tolerance, and the potency is much better than the bone marrow MSC, 3 to 5 times more, and also the validated robust GMP production by these cells. The production or expansion of this cell is much stronger than any other stromal cell. In fact, the DSC is not a better MSC. They are designed for the immunomodulatory. This is the recent data that we had in the Taskin. All of these patients were grade 4 SR-GVHD. 2 of them were even resistant to ruxolitinib. But after 1 to 4 doses, almost all of them reached to complete response, 2 of them to partial response. So the GVHD is the best model for the acute inflammatory disease because it mimics all of the TLPR1, TLPR17 pathway, endothelial damage, and loss of the Treg and alloreactive T cells. Therefore, we applied these cells in another acute GVHD, acute inflammatory disease like COVID-19. As you can see, all of the patients responded much better than the other in Iran and in Canada. And then the message is that multiple disease, acute inflammatory disease, or multiple disease, similar mechanism, but one transformatory treatment is a deciduous stromal cell, which are dedicated for immunomodulation. We published plenty of paper. We got the US FDA and the US and the world patent, as well as US FDA organ drug designation, Iranian market authorization, GMP certificate, as well as European orphan drug designation. We also got the US FDA IND approval for phase 2B clinical, which the project and proposal designing is moving forward. When you get for allogenic cell, when you get the US FDA approval, it means that the FDA approve your animal study, your human studies, cell toxicity, cell safety, phase 1, and then they gave us phase 2B clinical trial by the condition of the accelerated approval. The last and most important message, how we get to this point, is that following these 3R rules. These are the all human, normal human people. We collect the lymphocyte of them and stimulate them with a certain amount of the phytohemagglutinin. You see that this guy responded much more stronger than this guy. They are all of them healthy. The same mitogenic for all of them, the same conditioning. These 3 guys responded much stronger than these 2 guys. Certainly, we say that human beings are outbred. But here, we see that when we add one DSC, The lowest suppression was 43, but the highest was 80% suppression in the lymphocyte proliferation. It means that the human being, when we are using the cell therapy, are different and responded differently. If we want to use correctly, the best strategy as 3R rules, which says right disease, right cell, and right time. We should never forget that one stromal cell or one cell cannot be a magic bullet. We should categorize the patients or the diseases in the same category or same pathophysiology, and then we can use the cell which is fit with the underlying mechanism of the disease. Otherwise, we fail, like a lot of people which fail in the treating of the patient. The last thing is that the technical issue is very important, especially the viability and the other related factor to the cell safety and functionality. Thank you so much.